Hello, and welcome to an abbreviated overview of DNA replication. We'll start simple. This is a prokaryotic cell. This is what its DNA might look like. We'll assume it's a circle. And sometimes it just wants to divide as fast as it can. The DNA is usually anchored to the cell membrane, and two replisomes here begin their work. But then, if we take a step around, the newly replicated DNA also anchors itself to the membrane, and the cell elongates, pulling the strands apart as they separate. Before the cell has even finished dividing, its DNA begins replicating yet again, and so on. Now let's take a look at some of the finer details.
This is an origin site. It's where everything begins. This actually occurs in a massive protein complex called the initiation complex, but we'll ignore most of the tricky details and instead just focus on the essentials of replication. Here we have a few proteins working within and around a replication bubble, which is comprised of two replication forks extending in opposite directions. How DNA polymerase and primase orient themselves
depends on where the 3' and 5' ends are, since new nucleotides can only be attached onto the 3' end of the growing strand.
And can you see the symmetry here? Since DNA strands run anti-parallel to each other,
if you flip everything here 180 degrees, it'll look exactly the same. Okay, that's enough of that. For now, let's follow the left fork as it continues zooming off and... Oh, well, that's funny. It seems to have stopped. What could be happening here? Well, if we get a little closer, we'll find that the DNA has become much too twisted for the replisome to continue. So what now? Luckily, this is Topoisomerase, and it is tasked with unwinding the DNA. As we continue to push up the strand while straightening it out behind us, the front, well, it just keeps getting twistier. At this point, the enzyme breaks the phosphodiester bond, lets the whole thing spin around a bit, and then reseals the cut. Good as new. Helicase breaks the hydrogen bonds keeping the bases together, resulting in two strands. Single-stranded binding proteins then attach to ensure that they don't re-anneal before new complementary DNA has had a chance to be synthesized. Speaking of which, here comes DNA polymerase on the leading strand, and it's gone. Now let's take a closer look at that lagging strand, which, aside from its direction and length, is synthesized through exactly the same process as the other one. The resolution's a little low, so maybe if we... that's better.
In case you were wondering where the nucleotides came from, well, here they are, just floating around.
Primase lays down a few bases with NTP monomers. You know, the stuff that makes up RNA, before it moves on to bigger and better things.
Why did we need this?
Well, that's because DNA polymerase just isn't built to start on a single strand. It needs this open hydroxyl group here to begin doing its work. But when provided that, and a sliding clamp to keep it in place,
it can lay down dNTPs across long sequences with astonishing accuracy. This is due, in part, to its ability to sense the shape of and detach nucleotides that it has improperly paired, which is referred to as 3' to 5' exonuclease activity. This newly synthesized, smallish strand is known as an Okazaki fragment. DNA polymerase replaces the primer, and any missing phosphodiester bonds, like this one, are catalyzed by ligase. And there we go. Now we have two full double-stranded DNA molecules where there used to be only one. And they're pretty much identical to each other if we just ignore any mistakes that do inevitably crop up. In accordance with what is known as the semiconservative model of replication, half of each chromosome consists of newly made DNA, and the other half of previously existing template DNA. And now we can do this all over again. Forever.